I'd like to thank the Kidney Health Initiative for the opportunity today to present the work that we're doing in the Kidney Precision Medicine Project, or KPMP. This is an NIH, NIDDK funded consortium with the goal of finding breakthroughs in kidney health in order to benefit our patients. In many ways, the goals of the KPMP align extremely well with the goals of the Kidney Health Initiative, as we hope to develop tools that will allow for the development of new therapeutics that will benefit our patients. So for this presentation today, I'm going to start with a quote from Margaret Hamburg when she was the commissioner of the FDA and published this article in the New England Journal of Medicine, where she used very strong language for an FDA commissioner, saying we are failing as a scientific community and as a nation to adequately deliver the promise of science to diagnose, treat, prevent, or cure disease. And she said we must invest in new methods, assays, standards, and models that will help speed the development, review, and approval of medical products that patients need and can rely on. And really what the KPMP is trying to do is use new methods, assays, and standards to help that process along. And not long after that, Francis Collins, as the head of the NIH, also in the New England Journal, presented an initiative on precision medicine where he discussed the idea that one size doesn't always fit all when it comes to treating patients. And if we're going to take a syndrome such as chronic kidney disease or acute kidney injury, we have to realize that this is a heterogeneous syndrome and that the patients with those conditions differ from each other. We have to be able to find the right treatment at the right dose and get that to the right patient at the right time. And in order to do that, we'll need to be innovative and have novel designs as well for generating data and for clinical trials. And one could say that what the KPMP is trying to do is combine those visions, the visions of Margaret Hamburg and Francis Collins and Harold Varmus, and apply them to CKD and to AKI. So KPMP was conceived with the goals of identifying new treatments for kidney disease and to focus on both acute kidney injury and chronic kidney disease. And what makes KPMP unique is that we will ethically collect kidney biopsies from participants with these conditions who are altruistic individuals and will undergo a biopsy for research purposes and not necessarily because they themselves will benefit from the information that accrues from those biopsies, but for the greater good. And we'll use the tissues that are collected to create a kidney tissue atlas in health and disease, and to define within these syndromes of AKI and CKD subphenotypes or endophenotypes or different subgroups of patients for whom the disease process and the molecular drivers are different and the therapeutics should be different as well. So these are broad, ambitious goals of the KPMP. And this consortium, which is an ambitious consortium, now in its 6th year of funding from the NIH, consists of recruitment sites where participants are enrolled, recruited into this longitudinal cohort study and undergo a kidney biopsy as part of that process. There are so-called tissue interrogation sites that will use that kidney biopsy tissue and apply various kinds of state-of-the-art omics technologies to learn what can be learned from that tissue. There is an opportunity through what are called opportunity pool funding mechanisms to bring new technologies, new techniques into KPMP as the study evolves. There's a central hub that is the administrative core and the data coordinating center for KPMP. And there's the Kidney Mapping Atlas project, which takes these data that come from these biopsies and all of the orthogonal data elements that are collected in KPMP in order to create an atlas of the kidney in health and disease. One of the unique and distinguishing features, which we're quite proud of in KPMP, is the extent to which we walk the walk and not just talk the talk of patient engagement, and really have patients driving KPMP as equal partners in everything that we do. There is always a patient voting member of the steering committee. Patients are on every single committee of the KPMP. The initial draft of the informed consent was written by patients in our community engagement committee. Patients advocated for our obtaining no-fault harm insurance for participants in the study. And the community outreach is led by patients and community advisory boards that we're forming as well. And the committees with patients are really listened to carefully in the study. So we really do feel as though this is a patient-led study and really almost unique within these kinds of funded studies. What do we mean by an atlas? Well, there are a lot of different versions of an atlas, of course, going back half a millennium at this point in time. And certainly when I was growing up, we had a Rand McNally atlas. If you were driving in your car, that would tell you how to get from one place to another. And that has evolved over time. And now we think of the atlas in KPMP as being more like not just GPS, but more like software like Waze, where not only does it tell you how to get from point A to point B, but how to do that in the fastest time or the most direct route. And along the way, it's quite dynamic and living so that if there's a change, then it immediately adjusts to those changes. So we think of the KPMP Atlas as being really a dynamic, living, visual representation of all the different kinds of data domains that we're obtaining and linking those to what's going on in the kidney from a spatial perspective. This is a snapshot of what the study protocol is all about. After enrollment and recruitment into the study, when people meet the criteria for CKD or AKI for enrollment in the study, they undergo a kidney biopsy. That kidney biopsy tissue undergoes traditional pathology as well as a digital interpretation of the biopsy. As mentioned, the tissue is sent to the tissue interrogation sites for imaging and imaging analysis. The participants then are enrolled longitudinally, essentially in a cohort study. Clinical outcomes are tracked. Multiple data points, biosamples are collected, and all these data are used to create a kidney tissue atlas and to develop the goals of the study, mechanism-based disease subtypes, and also to identify the critical cell types and pathways and targets that will allow for novel therapy creation. An important aspect of KPMP is the data must be fair, which means findable, accessible, interoperable, and reusable. And that implies that we're going to integrate these data and that this is open data science, and we're going to make these data available to the entire community of stakeholders with an interest in kidney disease. So to make our data as useful as possible under fair constraints, we are combining digital pathology, the omics technologies, the clinical phenotypes together, and we've created ontologies, which are knowledge frameworks that allow data to be computable and to cross-compare between different studies, whether we're talking about apples to apples or oranges to oranges, and to integrate these data. And one of the unique aspects of KPMP is that as raw data is highly curated and QC'd, it then goes into the public domain as part of open data science, with the exception that if data would make participants identifiable, then one needs a data use agreement to use identifiable data. But all other data goes into the public domain as soon as it's really been QC'd, annotated, and curated. And we also provide tools on our website to use those data for various kinds of users who might want to use KPMP data. So starting from the bottom up, we have the initial data source, which comes from the recruitment sites, the tissue interrogation sites, the central hub, the different kinds of data go into a knowledge environment where we use ontology mapping, biocuration, extensive QC process to integrate these data into the atlas. And then we're creating personas of the different kinds of users and tools for those users so that the data will have maximal value, not just for researchers, but also for clinicians, for participants in the study, and for pathologists as well. So we had to create in the study a common vocabulary and new types of ontologies or knowledge frameworks. This effort is being led by Oliver He and Todd Valerius, and we've engaged other consortia in this as well. This is to benefit the entire field, to make data as useful as possible for anybody who wants to use the data going forward. And this is really open data science and collaborative science, and there are other Atlas efforts underway, such as the Chan Zuckerberg-funded Atlas, as well as HubMap, so the Human Cell Atlas and HubMap being NIH-funded across all organ systems. We work with these consortia, use common ontologies, common language, so that the data can all be used in the most effective way. A major goal of the consortium is to integrate the wealth of clinical data, structural data, and molecular data that we're obtaining in order to accomplish the goals of the study. This is a non-trivial process, and it involves really building a multimodal atlas of the human kidney in health and disease, with many, many different types of data that are being integrated together and then visually presented. Just as examples of what we're doing in terms of the biopsy data from both a histological or structural perspective, as well as the multi-omics kinds of data, I'll start by a description of how we're using both human analysis of biopsy data, as well as we're using machine learning and AI approaches for really a different way of quantitatively looking at biopsy tissue. So on the left-hand side, you can see what looks like traditional histology that we're all used to seeing from kidney biopsies. On the right-hand side, you see work from 3 different groups that are experts in machine learning and interpretation of histological images, and these 3 different groups are collaboratively working together, not competing, but working collaboratively in KPMP, both to obtain QC from a quantitative perspective on biopsy quality, then for tissue to undergo a segmentation process, where we basically can define borders around different structures, such as glomeruli, vessels, et cetera, within the kidney. And then also on a pixel by pixel basis, characterizing all the structures inside the kidney. So when we do this, kidney biopsy, as you can see on the right-hand side, looks nothing like what we're used to seeing over in the upper left-hand corner from our traditional nephropathology interpretation of morphology and kidney biopsies, and we can get exquisite detail on cellular and subcellular elements within the kidney biopsy tissue. And this has allowed us, with some of these machine learning approaches, to now look at parts of the kidney that we could never really even see before in kidney biopsies. Here, we're now able to look at peritubular capillaries from a quantitative perspective, as well as describing what's going on within the cells in terms of expression of proteins, et cetera. And these are the peritubular capillaries we've known for a long time are critical in that loss of peritubular capillaries almost always leads to progressive loss of kidney function and chronic kidney disease. Turning now to molecular interpretation, we have a variety of different tissue interrogation sites that are looking at the epigenome. We're also doing whole genome sequencing. So we have the genome, the epigenome, the transcriptome, the proteome, and the metabolome, as well as 3D imaging technologies. So orthogonal technologies being applied to the interpretation of these kidney biopsies. I'll give you a run-through of some of these technologies that, just to show the power of these different technologies. So we use single-cell RNA-seq and nuclear RNA-seq, led by University of San Diego, WashU St. Louis, and also University of Michigan. to identify cell types and states throughout the kidney. We also are spatially locating proteins within the kidney. So Vanderbilt University and Yale University are examples of some of the tissue interrogation sites that are imaging proteins within biopsy specimens. And you can get a sense of the incredible detail now, cellular detail that we can see, and also the components of those cells. So here looking at the different proteins and their expression in different components of the kidney. Here's a third different protein technique using multiplexed ion beam imaging from the University of Colorado, which is particularly useful for examining immune cells, whether resident immune cells in the kidney or cells that are migrating into the kidney in states of inflammation. So we can integrate all these data with the structural data and clinical data. So we can go all the way again from the genome all the way to small molecules or the metabolome and spatially locate different molecules within these kidney biopsy specimens. So what can we do with all these cells? Well, here's one of the first, I would say, seminal papers that's coming out from KPMP. This will be published next month in the journal Nature, where we examined 400,000 cells from 42 biopsies and mapped 1.2 million cell neighborhoods, predominantly using transcriptomic technologies for this effort. And we could define not just cell types, but I think this is very important, also now cell states. So now we can start to say for a given cell type, if it's injured, is it going through an adaptive or a maladaptive pathway, reparative pathway going forward? And as we do that, we can start to map what we call trajectories of reparative states. So once a cell is injured, is it going, what's the process like for going through an adaptive reparative pathway or a maladaptive pathway that's going to lead to fibrosis, scarring, and progressive loss of kidney function? We can also start to define neighborhoods of cells, so cell-cell interactions in an area, let's say, of active inflammation or an area of fibrosis within the kidney when other parts of the kidney are quite normal. So we're really now able to peer down in exquisite detail at what's going on in chronic kidney disease and in acute kidney injury, and we're able to map that, and then we will ultimately be able to map that to clinical outcomes as these participants are followed longitudinally. Now, we don't have the longitudinal follow-up because it's a relatively new study, but we can take already what we learned in KPMP and apply it to other cohort studies that exist. So we defined a thick ascending limb score for being activated and either adaptive or maladaptive. And we found that applied to other consortia, we've looked at Neptune and looked at several of the ERCB diabetic cohort as well. And these scores that we're seeing, that we're identifying for the first time in KPMP, turned out to be highly predictive of long-term outcomes in a diverse state of kidney diseases in these other consortia. So I've mentioned that this is open data science, and we're very excited about that. So you can go to the KPMP website, which you see listed on this slide, and there are now well over 4,000 dataset files that you can download and analyze. As mentioned, if you sign a data use agreement, you can get additional data that is potentially identifiable. That number is increasing exponentially as more and more data becomes available, QC'd and curated in KPMP. And already in a little over a year, there have been over 20,000 downloads of this data from around the world. So many different researchers, investigators, biotech, pharma companies are now using KPMP data. And that's a mark of success of the consortium. If you go on our website, you'll see a variety of different tools, and we're developing new tools all the time that can be used for working with KPMP data. the Atlas Explorer, the Atlas Repository, and the Spatial Viewer. And again, we are trying to put more and more tools on the website to make these data more and more usable by the entire community. And we're very open to collaboration. So the public data is available. One can submit ancillary studies to KPMP. You can sign a data use agreement to get protected data. And there are biopsy samples and materials that can be made available through an ancillary study process in KPMP. This is just a listing of some of the biosamples that we're collecting in this cohort study above and beyond the kidney biopsy tissue itself. And we welcome industry involvement, and we are planning, and of course, Kidney Health Initiative is a great group for us to get an opportunity to present what KPMP is all about, as we're doing today. And we want this to be bi-directional. We want the data that we are developing in KPMP to be useful for drug development, because the whole goal of KPMP is to have better therapeutics for our patients with CKD and with AKI. So we are planning in the fall an industry roundtable, and we welcome all of the members of KPMP to participate in this. More to come about this. We're hoping to tie this timing-wise to ASN Kidney Week so that it would be easy to attend, but we'll work on the date. But the goal is for us to be able to share what we're doing in KPMP, but also for industry to tell us, how is our data useful? And how is it maybe not as useful as it could be, and how can we make KPMP a better consortium? So I'll end by saying KPMP is unique in precision medicine. It really does involve an entire community. Patients are equitable partners, and we really, have a culture and a priority that's of safety for patients coming first. Many precision medicine studies really address either cancer or rare diseases, but here we're addressing the big public health problems, CKD and AKI. The Atlas has a clinical orientation as well. So we're very excited about this study, and we hope it will be of value to all the members of the Kidney Health Initiative. So I want to thank you for your attention. And we'll be happy to answer any questions. And please contact me at any point in time if you want to know more about KPMP.